Today I'll be presenting a novel virus hidden within the Tsetse Fly-Trypanosoma system called Narnavirus subdolus. As some background, tsetse flies are blood-feeding insects of the genus Glossina and are the vector for African trypanosomiasis, a potentially fatal disease endemic to sub-Saharan Africa. The disease is spread through the bite of a tsetse fly infected with trypanosoma. In animals, it's caused by subspecies such as Trypanosoma brucei, while in humans it's caused by subspecies such as Trypanosoma brucei gambians. In addition to these vector-parasite interactions, viruses have been discovered independently in both tsetse flies and trypanosomes, indicating that they may be a reservoir for currently unknown RNA viruses. It's important to investigate these possible viruses, as the fly-trypanosome system efficiently transmits disease. Narnaviruses are one group of RNA viruses that are typically around 3,000 nucleotides long, consisting primarily of a single open reading frame encoding an RDRP on the direct strand. This study will characterize a novel narnavirus in the Tsetse Fly-Trypanosome system and determine its likely host. Viral sequences were searched for within the Sequence Read archive, where the palmprint sequence of an RDRP was detected in 53 runs of the tissues of Glossina morsitans, 18 runs of Trypanosoma brucei, 2 runs of Trypanosoma congolens, and 1 run of Glossina papalis gambiensis. These came from 12 different bioprojects associated with transcriptomic studies that used Illumina sequencing at multiple institutions. BLAST-P analysis of the palmprint sequence showed homology to RDRPs of Narna-like viruses. Therefore, the sequence is hypothesized to represent a novel Narnavirus that infects either tsetse flies or their associated trypanosome parasites. The palmprint sequence was present in multiple short contigs across several runs. To extend these contigs, 3 datasets with the highest coverage were subsampled to adjust for memory limitations and assembled using megahit. A 772 nucleotide long contig was recovered from SRR 039378. The assembled viral contig was analyzed using ORF-Finder to identify protein coding regions consistent with Narnavirus genomes. The predicted open reading frame was characterized using NRPro and BLAST-P, revealing a conserved RDRP domain with homology to known Narnavirus and Narna-like virus RDRPs. Host association was assessed by comparing viral sequence coverage across sequencing datasets from different host organisms to infer active replication. An ORF spanning nucleotides 268 to 771 was identified in the contig. NRPro analysis revealed an RDRP domain characteristic of Narnaviruses at residues 8 to 131 of the translated ORF, or nucleotides 289 to 660 in the contig. Since this is much shorter than an expected Narnavirus genome, the sequence represents a fragment of the viral genome and was used in further analyses as a representative viral sequence. BLAST-P analysis of the translated ORF showed strong homology to Narna-like virus RDRPs, with the top hit sharing 70.8% identity with mid-Vegier chrysops, narnavirus-like virus 2. Phylogenetic analysis places the virus within insect-associated narnaviruses. However, sequencing coverage of the RDRP palmprint is much lower in Glossina morsitans, with the mean being 12.5, compared to Trypanosoma brucei, where the mean is 263.1. Suggesting that the virus is more likely replicating in Trypanosoma brucei, with detection in Glossina possibly reflecting contamination. The evidence supports the hypothesis that this is a Narnavirus infecting the Tsetse Fly-Trypanosoma system. However, there's a clear mismatch between phylogenetic placement and host abundance. Although the virus clusters with insect-associated Narnavirus lineages, sequencing coverage is much higher in Trypanosoma brucei. A similar mismatch has been reported for a Narna-like virus detected in Plasmodium vivax and its mosquito vector, where later experimental work showed the parasite was the true host. This suggests that in tightly linked vector-parasite systems, phylogeny alone might not be sufficient to accurately assign a viral host, and further experimental validation would be necessary. To conclude, Narnavirus subdolus is a newly identified Narnavirus, discovered through transcriptomic data. Phylogenetic analysis points towards an insect-associated virus, while sequencing coverage suggests a trypanosoma host. These results show how challenging host assignment can be in vector-parasite systems, and highlight the need for experimental validation to determine the true host.